Starting. There you go, that beautiful sound. Okay, so hello everyone. My name's Rudy, and I'm here to do a journal club review of this paper about the genetics of human aging. It's my first journal club review, so kind of nervous, but hopefully it'll be fine. So thank you again for coming, and if you're watching this virtually later, I guess thank you for watching. So this paper is unlike some of the other ones that people have done because it's a review instead of a sort of wet-based lab report type thing. So it's a review of a review in that sense. So if anything's a bit too vocabulary heavy for you, sorry about that. There are some statistics involved in this, although I don't really uh, know the minutia of it myself because I'm not a statistician by nature or practice. But if you're into that, there is more summary tables that were supplemented with this paper, so feel free to check them out, and I will link the reference below. So starting off with that, the genetics of human aging. So before we get into anything really heavy, I figured we could cover some basic definitions just used throughout the paper so that anyone that has confusion will hopefully clear that here and now. So first and foremost, aging, according to the scientists here, was used as a term to describe a correlated set of declines in functioning with advanced chronological age. And so this sort of decline begins to happen after humans or other animals reach their sexual maturity. And then linkage disequilibrium is a non-random association between alleles at different loci. And so it's a method for collecting data based on those sort of non-random associations. And it was the focus of some of their first GWAS studies. So next we have antagonistic pleiotropy. Sorry if I'm pronouncing any of these things wrong, not super familiar with the terminology, but this was a theory arguing that some mutations were selected because they're beneficial to early life fitness, although they can be harmful later in life. And so that is associated, therefore, with aging. And some examples of that include like muscle fitness proteins and other small sort of proteins like that. And then clonal expansion is just the production of daughter cells from a single parent cell, all sharing a similar particular trait that gets passed down. And then GWAS is a study that they drew upon for the vast majority of this review, and it involves genotyping a large number of people or models, whatever your sort of organism of study is, to identify any statistical associations between genetic variants. So on the topic of aging today, this was of particular difficulty because it's quite hard to pinpoint the phenotypes and genes involved with, I guess, aging because it is a multi-systemic process. And so that was something they'll talk about being a struggle later on in the paper. But just to keep that in mind, that the GWAS, although it's good because you look at this wide set of range, oftentimes the first obstacle of even picking the sort of genetic variant that you want to look at can be quite difficult. And so the question that the paper looked to answer today, is it possible to sustain longer lives with reduced periods of disability? And so this is something that's kind of a topic of interest for transcendentalists and people who, dream of freezing themselves after death in hopes of, I don't know, regenerating in a future time when science is improved. But to get to that point, I guess we have to answer these very basic questions, and genetics is a huge helper in doing that. And so some previous examples of slowing aging in laboratory models was demonstrated in C. elegans, mice essentially, and humans. And so in the C. elegans model, modulation of an insulin channel protein showed an increased health span. However, this came along with issues in fragility that was proportionally higher than the typical lifespan of a C. elegans worm. And so what that means is, although there was an extended period of time in which the C. elegans, thrived and was able to go about, I guess, life without any sort of health complications, the period towards the end of their life, they noticed a longer time at which there was just an overall weakness, not necessarily anything in particular wrong with the C. elegans, but just overall fragility throughout the organism. And so in particular with the C. elegans model, they noticed that DAF2, which is a specific nutrient sensing sort of growth factor associated with the insulin like growth factor 1, signals and upregulates autophagy and normal senses. And EAT2 is another sort of transcript factor that codes for a defective nicotinic sorry for the pronunciation, acetylcholine receptor, causing impaired pharyngeal pumping and genetic form of caloric restriction. And so these are all different factors that the DAF2 is the only one in particular that is involved with that insulin, IGF-1, but these other factors also played a role in affecting the lifespan and overall longevity of these different C. elegans. And so the last major one was a knockout of IFE2, which was known to increase lifespan by downregulating protein translation. And sorry, another last one, but knockdown of the clock gene CLK1 also affected longevity by impairing mitochondrial ATC activity. And so in their own small ways, the IFE2, CLK1, and EAT2 were able to affect the lifespan in a small way, but only the IGF-1 significantly increased the health span across multiple tests with the C. elegans. And so next, in mice, it was noticed that dietary restriction increased longevity, and this was in part because the modulation of IGF-1 and other growth factors were controlled by that sort of caloric restriction and micronutrient overload or regulation. And so this affected not only the IGF-1, but also mammalian target of rapamycin or mTOR and ribosomal protein S6 kinase, which is a signaling pathway also involved with growth factor control and regulation. And so the effectiveness of this dietary restriction is evolutionarily conserved across yeast, worms, flies, and other mouse strains, but it was observed to have more range in primate experiments, and that it wasn't always 100% effective in extending the health span and overall life frame of primates. And then finally, senescent cells in human is anything that is sort of responsible for the replication processes, secretion of cytokines, chemokines, and other growth factors that promote resistance to apoptosis. So in this case, targeting these cells kind of shut down these pathways, and meaning there was more cell damage inflicted, which overall affected the lifespan of the cells in particular. But a larger scale of this model is for the first time in trials. It's the first in man clinical trial in idiopathic pulmonary fibrosis. And so far, targeting these cells has been reported effective and relatively safe. So hopefully applications to other models that will then slow the age of those cells in humans will be effective in future as well. But at least we have some place to sort of start. And then we have, specifically for our purposes today, the approach was to use genetics to improve understanding of some underlying mechanisms of aging, specifically focused on loci previously identified through GWAS to involve human-specific aging traits, and prototypical progeria symptoms, so sort of the clinical manifestation of phenotypes that are already linked with aging malfunction and sort of quick aging, if that is the correct terminology for it, whether or not that could be applied to the larger human population based on the mutations identified in those specific syndromes. So for further clarity, progeria is any of several rare human disorders associated with premature aging, and the two major types are Hutchinson-Gilford and Werner syndrome. And so Werner syndrome manifests in adults, whereas Hutchinson-Gilford's can have its onset in early childhood. So our first figure for you today is the genetic overlap between age-related chronic diseases and parental longevity. And so this is based on correlations between GWAS results as well. So they identified some of the most common age-related diseases and wanted to put them in association with parental lifespan to look at the correlation between, I guess, age of onset and the overall lifespan of that said adult. And so genetic correlations here were gathered from the linkage disequilibrium score regression methods that were mentioned earlier in our definition section. And so statistically significant correlations were illustrated with an asterisk. So you can see, for example, chronic kidney disease has the double significance of the ones neighboring chronic kidney disease indicate that it was statistically significant through a Bonferroni correction, whereas a single asterisk just follows the standard p-test significance set to inequivalence, or not equivalence, set to a statistical score of 0.05 significance. And so the diseases in parental lifespans were ordered by similarity and patterns of genetic correlations using clustering, which is in one of the supplemental tables explained in further detail. So again, I can't really speak to the statistical minutia here, but if you look, you'll see that the orange or more red tone with negative correlation coefficients, I believe that meant that there was a less strong connection with the parental lifespan in terms of onset. And then the more positive ones had a stronger correlation, unless I've completely missed that in my interpretation. But would love to hear anyone's feedback if they have a better, I guess, grip on the analysis of this data. And then the next figure here covers the loci correlated variants associated with 3 or more age-related diseases or lifespans. I feel like for our purposes, this is a lot easier to digest as a reader because you can see the significance of the crossovers and things. And I guess if you're not colorblind, then the color coding really helped. But overall, there were 961 genome-wide significant variant trait associations across the selected age-related diseases, those all being previously covered in that other slide, so including things ranging from strokes to heart disease to cancers. And so based on the focus that the variants affect several age-related diseases, these were more likely to reveal, as we said, underlying aging mechanisms. They targeted loci associated with 3 or more and found 22 such multi-trait loci, of which 12 had variants in LD. And one of the loci in particular, APOE, has been associated with Alzheimer's disease and chronic artery disease, as well as with exceptional longevity and parental lifespan. So it's a sign that that loci, APOE again in particular, could be something of a target for clinical trials or further research in future. And 2 other loci, LPA and LDLR, contain variants that affect blood lipid levels and cardiovascular traits, which are also strongly connected with age-related diseases and longevity. And so those could also be targeted for future research as well. And then shown in this figure, specifically 9 loci were identified as hotspots for at least 3 major age-related diseases. And these were all done in a multi-correlated regression with over 0.6 genetic variants. Again, apologies, don't really know the statistical validity of that number there, but the lipid-related variants were excluded for simplicity of creating this table, but had they been added, there would have been a lot more crossovers. And the genes shown on the left, each with a link to the disease on the right, indicate a GWAS identified signal. So that right there shows the direct application from data gathered from these GWAS studies put into application in a table for visualization. And so the lifespan, as we've spoken about before, refers to a parental lifespan. So for example, a full-grown adult that was used for the collection of this data. APOE, sorry, like I said, is excluded on this picture, so that's why I don't see that there. Sorry if I miss any chat messages, by the way, just saw that one. So next we have overall genetics of clinical phenotypes. So more specifically, I guess, delving deeper into those already known diseases. So Werner Syndrome, like we said, is the adult onset version of one of the progeria, and it involves a mutation in the WRN gene. which encodes a member of the RECQ family of helicases that is involved in DNA recombination, replication, and telomere maintenance. And so that can kind of perhaps lend some insight into why this is adult onset, because it takes a significant aggregation of this mutation over time to appear in phenotype. Seeing another chat. Okay, sorry if I missed that. Okay, and then we have the Hutchinson-Gilford progeria, which is a heterozygous mutation in the LMNA gene. And so more than 90% of Hutchinson-Gilford progeria are caused by de novo heterozygous mutations. However, some others are involved in point mutations to exon 11 of the LMNA gene, which can disrupt production of progerin, which is a protein that influences fragility of the nuclear envelope. And so that breakdown can obviously have dramatic influences on the lifespan. And it ranges really with the different type of mutation and the expression levels of it. But this can obviously be life-threatening to a lot of children that have this disease. But more specifically, that 90% of heterozygous mutations manifest in disrupting proteins laminin A and laminin C through alternative splicing. Laminin A and C are otherwise assembled to form a matrix on the inner surface of the nuclear membrane. And the integrity, obviously, of the nuclear membrane is important for DNA maintenance mechanisms, such as, DNA double-strand breaks and other sorts of repairs. And so a disruption to this pathway can significantly disrupt DNA maintenance and protection. Finally, we have a syndrome that wasn't mentioned before, but Cockayne syndrome, which is caused by mutations to the ERCC8 gene and ERCC6 gene, and these genes, encode the CSA and CSB proteins, which play central roles in the transcription-coupled nucleotide excision repair, which occurs at the specific secondary DNA structures. And so this can have a range of different targets, but all manifest in some way to affect the longevity of the patient or person that has it. And so to conclude about these specific clinical phenotypes and their values, the data on progerias indicates that mechanisms of DNA repair and maintenance are super important for health conservation, and whether these are directly connected or underlying true accelerated aging syndromes isn't necessarily clear yet. But a key question that follows is whether or not these generally common variants mapped to these same progeria genes are important for the common traits in the general population. So basically, it's still uncertain as to whether or not these sort of mutations can generally affect people, or if it's only in these specific sort of syndromes where they play that direct role. But future research will show that and potentially lend insight into treating the general population, and maybe there's a positive mutation that we can influence on these genes that would have some sort of reverse positive effect, but I guess we'll see. So it should be noted as well that at this point in the paper, further discussion went on to cover somatic mutations and their sort of involvement in, again, influencing aging and things, but it's such a complicated process that I decided to exclude that from our discussion today. But it could be noted that lots of it was prevalent in women that they studied, and specifically prevalent at the age of menopause. So that's quite later on in life. So the question remains then whether or not some sort of advancement to extend the lifespan could come in and, I guess, prevent some of those things before menopause, or if it would be better to target and do whether or not it would be better to take a preventative or reactive approach, and which of those would be effective and at what age points we should interfere for that. But those are just future questions that we have. But if you're more interested to understand all the somatic mutations, feel free to delve deeper into this paper again. It's a long one, but it's very interesting and has lots of applications we can look at for future, I guess, directions for aging research. And so next, this is just to provide a little background on some previous evolutionary theories of aging. So the first one you have here is early group selection. There's not necessarily a specific person to credit this theory to, but it was a generally shared belief that aging is a genetically encoded adaptive trait and increases mortality in individuals with declining reproductive potential, therefore freeing up resources for the younger population or generation to reproduce. And so this theory was abandoned by the sort of original group of subscribers because evidence that natural selection is most effective at the individual level, even when it conflicts with sort of the species survival as a whole, is yeah, it's conflicting there because obviously, an individual will still try to thrive and live normally in their habitat without consideration of the species. And so additionally with that, no organism level programmed death gene has been found. So there's not one ticking time bomb among the population that will sort of wipe out a whole age group just to maintain the survival of younger generations. But the other most accepted theories are the ones below. And so starting with that, we have the mutation accumulation theory. And so this proposes that organisms accumulate damaging germline mutations expressed only in the post-reproductive period of life because these mutations would not be eliminated by earlier selective pressures. So when you're kind of growing and developing as a kid, there isn't really the space or, I guess, drive for mutations to get eliminated. And so this theory interprets variations in lifespan between different mammal species as related to the differences in age of sexual maturity. So for humans, Huntington disease provides probably the best example of that. But in the post-reproduction, increase in death rates has not been detected in humans or animal species. So that is to say, rapid death isn't necessarily that would be an extreme to draw from this theory, but it isn't necessarily found in practice or in nature. So again, that's why it's a theory, not a commonly accepted fact, I guess. But then we have the antagonistic pleiotropy theory, which argues that some mutations are selected because they're beneficial to early fitness but become harmful later in life. So this is something we covered in our first definition. And this sort of selection of mutations that are beneficial and then harmful is what causes aging. So cell senescence pathways are the best example for this, which is a programmed senescence that occurs during normal mammalian development, protecting against cancer and wound healing at younger ages, but then contributes to degenerative chronic diseases at older ages. And so this theory has lots of applications. It's very beneficial for the GWAS studies that were performed here. And so whether or not you subscribe to that, interesting, but something to consider. And so then we have, last but not least, the disposable Soma theory, which states that given the availability of limited resources, aging arises from evolutionary trade-offs between growth and reproduction. So that is on one hand attributed to that, but on the other hand attributed to repair mechanisms. So it's consistent kind of with evidence that long-lived species with kind of robust histories such as humans evolved by developing more sophisticated and effective repair mechanisms. For example, the comparative studies that were reviewed by this paper have found the capacity to recycle deteriorated macromolecules and organelles through autophagy correlates with lifespan. And so that is just one evolutionary component that could perhaps contribute to our aging determination. And so with that, human resilience mechanisms were selected to be robust enough to match the environmental pressures that drove human evolution. And recent dramatic decline of environmental pressures, though, has extended survival beyond the previous resilience warranty period. And so now we are at a point in time where contemporary environments allow for a more lengthy survival after the reproductive period, with an eventual increasing damage over repair towards the end of life. And so that is something that is attributed with sort of our lifestyle diseases and things. But yeah, fitness has declined in some senses because of the way that humans live their life now, more sedentary and things. Sorry, dog needs rubbing. Oh my God. But yeah, so this sort of disposable soma theory will have interesting future applications as well as that sort of contemporary environment period extends. And so finally, something to consider when doing these sort of studies on aging is that there's a current lack of standard terminology. It's hard to distinguish between the causal and determining factors, and there are variations in study design to a point that results almost aren't comparable and they can't necessarily build off of each other. And as with any study, there's also ethical qualms. Obviously, a sort of wet lab, hands-on molecular view at aging would be very invasive, and it would have to follow perhaps people throughout their whole lifetimes. And you'd have to do that with a large group of people for, accuracy and everything. So there are ethics qualms, but not to my knowledge have there been any studies, I guess, applied for to that effect or extent. Um, sort of like I'm sure there have been in some smaller scopes, but I don't think any labs have proposed a way to study people for that long and what they would hope to achieve from that sort of a study. But then finally, the range of phenotypes with potential influence on aging. So this is an issue in our study today. They were able to focus on, some of the big loci like APOE and LMNA, but there are so many potential factors that we don't even know about that could play a role in our aging and, cell fate determination and things. So it's something that for GWAS style studies has to be improved upon, is selecting that sort of gene and then looking at the phenotypes as a result of that. But yeah, hopefully this will improve over time with the improvement of understanding about this. And then finally, we have, I guess, our reference. So this is where the paper came from. They did a really good job, a really comprehensive review of the current knowledge of aging and future approaches for it. But yeah, so that is all I have. So thank you guys for that. Sorry if there were a lot of random words. I didn't know half of them either.
Right. So thank you for Rudy's presentation. Are there any questions from the audience, or if not, we can just move on to the next one. Okay, so I'll just start sharing screen. I actually just did the presentation like literally today, because this paper is like published 2 days ago, like literally on November the 8th. And it's pretty interesting. I was originally planning on doing a bioelectricity thing, but then I saw this paper and I was like, no, I'm gonna do this. So yeah, just gonna share screen. Can you guys see the presentation?
No, not yet. Oh, yes.
Okay, right.
Perfect.
Okay, so this paper is talking about the a new kind of histone modification. So it's called histone H3 especially, and the 16th residue, which is a proline. It undergoes hydroxylation, and this kind of hydroxylation has regulatory effects on gene expression. Basically, yeah, so histone post-translational modifications, they could lead to a lot of different epigenetic regulations. The most well-known these kind of modifications would be acetylation of histones. That's, I think, is discovered in the 1960s or something. Another very common modification that we're actually going to focus on in this paper, in league with this newly discovered hydroxylation, would be methylation. And specifically, we're talking about the trimethylation of lysine 4 on histone H3 subunits. So I've attached like this diagram showing the histone structure here. Basically speaking, the blue subunits here would be the histone 3. That's on a nucleosome. And yeah, that's how they package the DNA. Anyway, so basically what H3K4ME3 does, that means the trimethylation does, is to make the DNA more accessible, because it interacts with the nucleosome remodeling factor, which is some kind of chromatin remodeling factor that will catalyze the nucleosome sliding. It basically makes more DNA exposed to and enable the binding of RNA polymerases and activates transcription of these genes. Like basically, there are different enzymes and proteins which will interact with these trimethylation sites. The one we will be focusing on is KDM5A to D, and especially on KDM5A for reasons we'll discuss later. Basically, what these readers does is that it demethylates these methylated states, and this will lead to a variable effects on gene regulation. So it can activate genes, or it can also repress genes. So these post-translational modifications as a whole, they can act as docking sites for chromatin modulators. An example would be this nerve docking site provided by the histone trimethylation. The novel PTM we're focusing on today is the proline 16 hydroxylation, H3P16OH in short. Proline hydroxylation is actually quite common, a PTM. It's actually one of the most common PTMs, especially with regards to histones. But then I had not have time to go over that. If anybody knows anything about this, just feel free to like supplement this. The paper did mention that it's quite challenging to assess proline hydroxylation, and so basically proline hydroxylation is not really commonly studied because of technical reasons, and basically known roles proline hydroxylation plays on the different proteins kind of metabolism effects would be oxygen sensing. So basically EGLN here, sorry, I should have let me just skip to the next slide first. Yes, where's EGLN? Right, so EGLN is proline hydroxylase, right? So EGLN is a proline hydroxylase, which will, of course, catalyze the hydroxylation of prolines on proteins. And if EGLN is under a normal oxygen level, it will hydroxylate hypoxia-inducible factors. And then this will lead to ultimately the degradation of this HIF. And then if it's degraded, there wouldn't be anything happening after the transcription of the factor. However, under hypoxic conditions, EGLN would be deactivated, mechanisms for which we'll go into later. And then the HIF1 alpha will not be degraded, and this stable alpha protein will bind to the beta protein, and this complex will promote a series of enhanced metabolism and repair mechanisms in the cell, and this will help cells to adapt to hypoxic conditions. Yeah, so basically there has also been other known roles of proline hydroxylation in cells, and one of them is actually gene regulation. So similar to what we've discussed earlier, which is the focus of this paper, basically the paper is saying that the hydroxylation of proline 16 on histone 3 is correlated with the demethylation of this lysine 4 on histone 3. So now there is another known kind of analogous situation happening that is previously studied, where the proline residue 38, upon being hydroxylated, it will enhance the demethylation of these kind of lysine 36. So you can notice some kind of like a linkage between proline hydroxylation and lysine methylation. Yeah. So the cell strain, like the cell line used in this study, is from the triple negative breast cancer, and it's actually the most lethal subtype of breast cancer. Reasons because it lacks all three receptors typically found on like the breast cancer cells, that would be progesterone, estrogen, and the human epidermal growth factor. This basically means that hormone therapy and other specifically targeting drugs cannot be used, and instead mostly people will rely on chemotherapy to attempt to kill off these cancer cells. And as we all know, chemotherapy is quite destructive. Right, so the correlation between, right, actually I have to like explain the background first. There's a lot of background here. TNBC earlier is actually triple negative breast cancer, right? So basically, upon this is like a canonical pathway, and then in the canonical pathway, the Wnt factor is upregulated in these cancer cells in the early outgrowth stage, and then this will lead to downstream reactions, which will upregulate the gene that produces beta-catenin. And then these kind of beta-catenins will accumulate in the cell. And then this will lead to, of course, by that time, we can already diagnose the cancer because there's a lot of beta-catenin, but then it will also lead to a lot of adverse effects. So another side note is that usually triple negative breast cancer, TNBC, has a low poor prognosis. It's hard to be diagnosed at an early stage because the Wnt factor is actually downregulated during dormancy. That means like during the early stages. And this downregulation can help it to avoid immune surveillance, but that also means that we don't really know if you've got the cancer or not until like later stages when the cancer is like spreading. And there is common problems associated with the treatment of triple negative breast cancer, in that usually nowadays the novel kind of like therapeutic measures would be to introduce Wnt inhibitors, which will inhibit the Wnt expression, and then this will, of course, reduce the beta-catenin production amongst any other things. However, the inhibitors of Wnt will also lead to the quiescence of the cancer cells, and because they're not really growing that much, they're insensitive to drugs. And this insensitivity to drugs means that you need to use more drugs, and you also need to continue to like administer these Wnt inhibitors to the breast cancer patients, so that the cancer cells, being practically immortal because you can't kill them with the drugs now that they're insensitive, so that the cancer cells will be kept at a quiescence level and the tumor will not proliferate. And because of this problem, the Wnt inhibitors, although there are actually a lot in the market right now, they are not actually commercially sold, especially not adopted in like many countries due to these quiescence reasons. Right, basically what happens in this, what happens is that EGLN2, the proline hydroxylase, will attempt to hydroxylate proline residue 16, the kind of like the PTM we're focusing on this time. And then this hydroxylation will recruit KDM5A. KDM5A is an enzyme that will demethylate lysine. And then this KDM5A will demethylate lysine 4, also on histone 3. And then after demethylation, this will lead to the DKK1 transcription being repressed. And DKK1 is actually an inhibitor of the beta-catenin production. Yeah. So if DKK1 is repressed, then more beta-catenin is expressed. And this will lead to, of course, more adverse symptoms of the cancer. So yeah, this is during the Wnt on stage. That means Wnt is kind of expressed. And when Wnt is expressed, as we've talked about previously, it's actually during the diagnostic stage, which is for triple negative breast cancer patients towards the later stage of their cancer. Yeah, so to delineate this, just go back to here. Yeah, to delineate this histone H3 proline 16 hydroxylation. So first of all, they need to know which EGLN, because we have 3 EGLNs, which EGLN will actually bind to H3 and hydroxylate H3. And this is because, one, they don't know which residue is being hydroxylated at the beginning of the study. 2, they don't know which EGLN is actually responsible for this hydroxylation. So first of all, they do this pull down assay to delineate which EGLN is actually responsible. And they found that only EGLN2, usually localized in the nucleus, will actually bind to H3. That means they can narrow down their scope of investigation on the proline hydroxylation to EGLN2. And then we can see that they did an immunoprecipitation step, and they also did a mass spectroscopy. So first of all, after finding out that EGLN2 actually interacts and binds to H3, we have to find whether H3 is subject to proline hydroxylation. And they do that because they don't know which proline is hydroxylated at this stage. The H3 histone has 6 prolines. So they need to do this by a pan-prolyl hydroxylase immunoprecipitation. And this will basically pull down all of the hydroxylated H3. And then, of course, they found that H3 is hydroxylated. And then, after they found that H3 is being hydroxylated, they want to find out, is the hydroxylation due to EGLN2, or is it because of other things they haven't investigated? Basically, what they did was to add DMOG, an inhibitor of prolyl hydrolases, and EGLN2 is also a prolyl hydroxylase, so DMOG will inhibit its action. What happened was that DMOG reduces hydroxylation of the H3 histone, but then if there's no DMOG present, the H3 histones hydroxylation level is higher. So that means the H3 histones hydroxylation is due to the action of EGLN2. And then afterwards, they need to know whether EGLN2 indirectly or directly causes the hydroxylation. So they did some other hydroxylation assays and find that EGLN2 consistently promotes hydroxylation of H3 histones. And then afterwards, they need to confirm the in vitro hydroxylation because as you've probably noticed before, they're using immunoprecipitation as a method, pan-immunoprecipitation actually, to determine whether EGLN2 is hydroxylated. And we all know that there might be some issues regarding non-specificity in immunoprecipitation, especially when this antibody is kind of a pan, like a widely applicable antibody. So it's not especially specific. We need to confirm whether H3 is really hydroxylated. So after they did all this confirmation, they also used mass spectroscopy to detect hydroxylation of H3 in transfected cells. So they confirmed that this process actually occurs in living systems. So MYC-HC just means that the HC is a recombinant protein tagged with the MYC tag. it's useful for purification. And then specific anti-H3P16OH antibodies are made, and this is from rabbit IgG cells, and they did dot blotting to confirm that the antibody is indeed specific. So this antibody is obviously used in the next steps. Histone marks are then analyzed, and then they found that the trimethylation of lysine 4 increases after EGLN2 is removed from the system. It's actually because, because they added siRNA to inhibit the translation of EGLN2. siRNA basically stops, they kind of degrade EGLN2 transcripts. And then they also added DMOG, which again inhibits EGLN2, and we can see that the H3K4ME3, that means the trimethylation rate is increased. What that means is that prolyl hydroxylation on proline 16 actually regulates this trimethylation on lysine 4, and the regulation comes in the form of it decreases trimethylation. So then they need to, what's there an H here? Anyway, they did a pull down assay and immunoprecipitation to find out whether H3P16OH, like the mechanism by which it decreases H3K4ME3. And the pull down assay basically finds that in the dual kind of PTM situation where the H3 contains both PTMs of trimethylation of lysine 4 and hydroxylation of proline 16, this dual modified histone will bind stronger to KDM5A. KDM5A, as I've previously mentioned, is the demethylase, which will demethylate the trimethylated histones. And they then found out that the proline 16 hydroxylated version of the H3 histone in isolation also binds to KDM5A. So then they can conclude that the hydroxylated proline 16 actually helps in recruiting KDM5A. And of course, because it helps in recruiting KDM5A, it will enhance the demethylation action. And this further decreases the trimethylated lysine 4 found on the histones. And then they then investigated which of the finger domains binds to the histone and found that it is actually PHD3, which binds to the histone. And then basically they did a co-immunoprecipitation of KDM5A with the hydroxylated proline version of histones. And then we can see that the KDM5A is efficiently co-immunoprecipitated with the hydroxylated proline histone. But then if there's like hypoxia, you might, actually it would help if I go back to the slides. Yep. Here, okay. So you might recall that hypoxia will deactivate EGLN, and then deactivating EGLN means that, yes, here it is. Deactivating EGLN means that H3P16OH is it is not hydroxylated, and then if it is not hydroxylated, it will decrease the trimethylation rates of lysine 4 residue. And then basically they found, they kind of like used this to prove their conclusion that the hydroxylation of proline 16 recruits KDM5A and decreases the trimethylation of lysine 4. And then they did some other things to see this, whether this occurs in kind of like a in vitro or like in a system, a living system. Basically, they did cut and run. I'm not really familiar with cut and run myself, but basically, this is a kind of like a immunoprecipitation technique. Yeah, I'm not an expert, so I won't attempt to like explain this. Basically, what happened was they found that EGLN2 depletion lead to notably decreased hydroxylation levels of proline 16, and that effect is not enhanced by EGLN2 depletion. So that means it's actually EGLN2 inhibition, which plays into the effect of what we noticed during hypoxia. And then they also did like RNA sequencing for the genes identified to bind directly to EGLN2 and all the things we've talked about that are involved in this mechanism. And they also, of course, identify genes that are negatively regulated by EGLN2 and KDM5. And yeah. Oh, do I open the chat? Right. Sorry. Give me a second. Actually, would you mind if you just ask in the, because I can't really see the chat here. I don't know why.
Okay, I'll just ask this way.
Yeah.
What is the point of the RNA-seq? Did they not know what targets there were beforehand?
No, they did not. So basically, they were just hunting down this hydroxylation PTM. They found it interesting, and then they wanted to like find what exactly does it do. So their main, they were like going after a threat. So somebody previously did hydroxylation experiments on histones, proline hydroxylation. And then they were like, okay, let's find other like proline hydroxylation sites. And then they found another one. And then now they want to like know whether what exactly does this do, and then what genes will it interact with. And then they found that it actually interacts with DKK1. And then, yeah, let me just go back to the, yeah.
So for all of those genes, they're all novel. No one's ever found them before?
They are known genes, but then they're not known to interact specifically with hydroxylated H3 at proline 16, because obviously no one ever found that the proline 16 residue could be hydroxylated. So I guess they're like the first ones doing that, and they actually isolated DKK1 in their evaluation section because of its like implications to the treatment of the breast cancer. So yeah.
Okay, cool.
I mean, they actually have like a list of genes, and I guess I shouldn't like talk about the whole list of genes because that's like a lot. Yeah. Okay. And then, so did I answer your question tangentially or?
Yeah, that's fine.
Okay. Anyway, you can see that the EGLN2 it interacts with the hydroxylated P16 of H3, and then this will recruit KDM5A. And then all in all, this interaction will regulate gene expression in mammals. And we don't know about other eukaryotes yet. So there's like further rooms for study. Of course, the whole purpose of this paper is to say that this kind of like isolated EGLN2 inhibitor is actually a valid candidate for treatment of the triple negative breast cancer. This is because, as we've said before, the Wnt inhibitor will turn off Wnt, and this will cause cell quiescence, and this will cause resistance to treatment drugs, and this is bad. So if we instead attempts to inhibit EGLN, this will not turn off Wnt. And then that means the cancer cells are still susceptible to drug treatment, but then beta carotene wouldn't be produced. So I guess the symptoms are less bad, and it also doesn't proliferate that much. And then it also lifts this is because it lifts the repression of DKK1, which inhibits the production of beta catenin. And however, this potential inhibitor treatment will only work on canonical pathways. What we mean by canonical pathways is that they are kind of beta catenin independent. You can see that basically there are currently 6 kind of like pathways introduced or discovered, and then you can see that pathways A, B, and D, they all contain beta catenin. It's kind of like a cofactor for that. But C also contains beta catenin in the diagram, but you can see like E and F is like the non-canonical pathways. And honestly, if you use EGLN2 inhibitors to treat ENF, there wouldn't be much results. So yeah, that for ENF patients, they might have to like stick to chemotherapy, right? I noticed that there's like a lot of terms. So would it help if I go back to like the flow chart here? Yeah. So that's basically what happens. If they inhibit EGLN2, this will stop this kind of like recruitment of KDM5A. And then you can see also that KDM5A also naturally just demethylates K4 of the histone H3, but then it occurs at like a slower rate, and then it's basically like not significant to the overall level of expression. So if they keep the Wnt expression on and then they inhibit EGLN2, this will not cause cell quiescence, but this will also effectively restore the expression of DKK1, the tumor suppressor gene. Yeah. Are there like any other questions, or would anybody like to like see other diagrams? Right. Yeah.
Sorry, just one question. So they've uncovered this part of the pathway. Is there anything more that they would want to uncover from this specific part of the pathway, or is it more now going to be research directed on application in clinical practice?
I think they were definitely pointing towards application. And I think they were also pointing towards like, more studies on like the hydroxylation of proline residues in general. They did mention that like they started off with a previous study of somebody doing similar things, and then they were just saying that maybe other people should like also do this. But I don't think they mentioned like anything further streamlining things. It might be on the other genes they discovered though. Like you remember like there were like an array of genes that actually kind of like tangentially interact, or like their regulation, like their expression is kind of affected by this whole system. So apart from DKK1, which is obviously a therapeutic target, there might be other genes that are actually of interest that might be worth looking into. Yeah. You can actually find this on if you type like Nature Genetics, it will appear like on the first page or something because it's a recent article. So yeah. And I'm also not sure about cut and run. So I don't know if anyone can like explain that. But if not, I guess if nobody has like any other questions, we can, yeah, end this.
I think I just one question. So what made you pick this paper?
Yeah, so because I was in a epigenetics lab previously. Like I was working as kind of like an intern, and they were studying histone demethylation, I remember. Of course, I was a high schooler, so I didn't know much about the specifics of what it means, but I just saw like this whole neo modification that someone just discovered, and I was like, yeah, maybe I should like try to do this. But yeah, I still have to like obviously put some more time into this, especially there were like some other experiments that I kind of skipped because they were proving kind of like circular things. I can actually show some examples of that. Wait. Yeah, so they did like, apart from cut and run, they did microchip, and then they also did like RT-PCR. And there was like this whole session on this grade analysis, which obviously, yeah, I would love some input from the audience if someone knows what it actually is. Yeah.
No idea, but it's all super interesting.
Right. Anyway, if there's like no questions, we can like end the journal club. If anybody would like to discuss this, or anybody knows like more things about it, just it would be great if you can like share or something, like we can discuss afterwards, because I'm also kind of confused on some of the specific figures they did. Yeah. Right. Thank you.
Awesome. Well, I'll stop the recording there. Thank